Okay, so this is going to be an extension of what we talked about in lab. I did spend about 20 or 30 minutes priming you guys with a lecture in lab. And so the first part of this lecture is going to cover many of the same, very similar concepts that we talked about in lab. Obviously, we're going to go into a little more detail here in lecture. And I'm going to attempt to get through most of the relevant material from chapter 12. This is one of those lectures where it is going to be impossible for me to cover every single concept that you're going to encounter, and it's just going to be very critical for you guys to make sure that you thoroughly read this chapter and make sure that you're good on those concepts, because I'm not going to be able to cover every single word, every single phrase, every single thing. So I'm going to hit the big picture material. Okay, I'm going to cover the most important, most relevant points, or points that I think are most important, most relevant. And we're also going to talk about some clinical applications to those as well. Like how can we use this information to help us as far as taking care of patients and problems that can occur. So, as we talked about in the lab, the nervous system, the basis of our consciousness, and again, we don't have a robust theory of consciousness. We don't have a robust theory, many ideas, many hypotheses, and we certainly know more now than we ever have. When I took anatomy and physiology back in the 90s, the understanding was significantly less back then than it is now, and the advent of new and improved technology has really helped us access neurology. We've made leaps and bounds in understanding what's going on. One of the most important techniques is something called fMRI. Have you guys ever heard of that? fMRI. It stands for Functional Magnetic Resonance Imaging. And magnetic resonance imaging is a very advanced way of viewing into the body. Typically we use x-rays, and I've talked about x-rays before in this class, but MRI uses, hi, how are you doing?
Good.
A little late. Magnetic resonance imaging is not an x-ray. It doesn't use x-rays to look into the human body. But rather what it does is it applies a magnetic field, and then that magnetic field alters the states of certain electrons, specifically electrons that are not paired up, what we call unpaired electrons. And as you guys have learned in chemistry, electrons have a property associated with them called spin. Right? You guys remember, you guys have all taken chemistry, I presume, you should have, or should be taking it to be in this course. So you know that electrons have a quantum state, what they call a spin state, and the spin can be up or down, or plus 1/2 or minus 1/2. And what we can do is we can use magnetic fields to kind of change the spin state of unpaired electrons, specifically unpaired electrons of hydrogen atoms. And what that can do, and then what we can do is we can kind of shoot radio waves in there, and we can kind of perturb how that spin is. And then what happens is as the electrons go back to that original spin state, they release radio waves, and those radio waves can then be detected, and we can use that to produce an image. Functional MRI is really good at looking at areas of the body that are more or less active. And so what we've done is we've taken people, we've put them into MRI scanners, and then we've had them think about certain things, we've had them do certain tasks, we have had them experience certain feelings and thoughts and emotions. And then what we can do is we can actually see what areas of the brain are more active. What areas of the brain are active when somebody is thinking or doing something. And what we've been able to do is we've been able to identify, okay, where in the brain is this thought occurring? Where is this emotion coming from? Where is this movement coming from? We map all that onto the brain. And then what we do is we look at the brain and we look at the tissues. Okay, what specific tissues are there? What specific cells are there? And what specific neurotransmitters and receptors for neurotransmitters are going on there? And by doing this, we've been able to piece together a picture of what's going on with the brain. But before we can look at the big picture, we have to look at the small picture. And so we're going to start at a nervous tissue. And as I've talked about in the lab, you have about 100 billion neurons in your brain. Okay, so you have about 100 billion neurons, or 100 billion stars in our galaxy, in the Milky Way galaxy. So, I mean, just imagine that. You have a neuron for about every star that there is in our galaxy. And then those neurons have thousands of different connections between, so each of them have thousands of connections. And so 100 billion neurons, and each with thousands of connections. And as you could imagine, how many connections are we dealing with? How many synapses are we dealing with? And then now we know that's not even the end of the story, is it? Because not only do we have neurons, but we have these glial cells, and these glial cells are very important as far as supporting the neurons and helping the neurons function. So it's a really complex picture, and we're only going to be able to scratch the surface in this particular class. So let's just talk about the nervous system real quick. So the nervous system has two major branches. It has the central nervous system and the peripheral nervous system. And the central nervous system is a part of the nervous system that exists within the axial skeleton, right? So that includes the brain and the spinal cord. The brain and the spinal cord is considered the central nervous system. And then all of the neurons that come out or into the central nervous system comprise the peripheral nervous system, or the PNS. So the CNS is the central nervous system, the peripheral nervous system is the PNS. And there are basically two ways to get in or out of the central nervous system. Okay, there are two ways of doing it. Okay, the first way is by going directly out of the brain. Yeah, right? There are two ways to get in and out of the central nervous system. The first way is direct. It is a nerve that goes in or directly into or directly out of the brain. Okay, so it's a nerve that connects directly to the brain. Those nerves are what we call cranial nerves. Cranial nerves are nerves that come directly out or go directly into the brain. And we'll get to, there are some nerves that go in and there are some nerves that go out. And nerves going in carry different information than nerves going out. The other way of leaving the central nervous system is by the spinal cord. You can have nerves that come off of the spinal cord, and we call those spinal nerves. So cranial nerves are nerves that come directly in or out of the brain, and spinal nerves are nerves that come in or out of the spinal cord. Does that kind of make sense? Okay, so those are the two ways in or out of the brain. Now, when we talk about in or out of the brain, okay, we need to understand that different types of information, okay, are carried into the brain, and different types of information come out of the brain. Okay, so in general, nerves that go into the central nervous system, that go in, okay, so I think of this as ascending, okay, because that information has to go up to the brain, right? Those nervous pathways that ascend, that carry information up to the brain, are what are known as afferent pathways. These are pathways that carry information into the brain, and that information may go into the brain through either spinal nerves or cranial nerves. And then very special, well, no, not even that, because vision, and as we'll learn, vision and hearing and all that is actually the result of cranial nerves. But, okay, so information coming into the brain comes in through afferent or ascending pathways. These are also known as sensory pathways. So sensory information ascends and goes into the brain. Does that make sense? Or ascends and goes into the central nervous system. Okay? Now, information that comes out of the brain is what we call descending, and it comes out through descending pathways. And primarily, information that comes out of descending pathways are also known as efferent pathways. So afferent is ascend, sensory. Efferent is descending, that is information leaving the brain. And that is primarily going to be motor information. Okay? Does that kind of make sense? So sensory information ascends, motor information descends, comes out of the brain, right? So sensory information comes in, the brain integrates that information, and then it sends motor information out in response to that sensory information. Does that make sense? So sensory information may be you see something on the table. Okay, it's something really tasty looking. It looks tasty, it's colorful, you can smell it. It smells really good. All that information comes in, and then your brain integrates it and says, "Oh, you better eat that." And then it sends motor information out and tells you to grab it and stuff it down your throat and swallow it, and you're good to go, right? Okay, so there you are. Does that kind of make sense? Now, with that in mind, the peripheral nervous system is further subdivided into two categories, if you will. Okay? So the CNS and then the PNS, but the PNS has two divisions to it, two major divisions. Okay? So the PNS has what's known as the autonomic division, or the ANS, the autonomic nervous system, and the somatic division. Okay? So autonomic and somatic are the two major branches of the peripheral nervous system. The autonomic branch, I think of autonomic as being automatic. Okay, so this is the involuntary branch. So this is information, okay? This is motor and sensory information, okay? That tells your heart to beat, that makes you breathe, okay? That tells your stomach to make more stomach acid, tells your intestines, the smooth muscle in your intestines to move and create those peristaltic motions that move food and chime around. Okay, this is all involuntary. Okay, we don't typically have conscious control over this for the most part. Okay, that's known as autonomic. And then the other branch is the somatic. Okay, and soma, remember, means body. And the somatic branch is the voluntary branch. It is the branch that we have conscious control over. Okay, so this is primarily going to involve skeletal muscle. You guys cool with that? Okay, so far so good. So autonomic, automatic, involuntary, somatic, voluntary for the most part. Now to even make it more complicated, the autonomic nervous system is further subdivided into two branches. Okay? The sympathetic and the parasympathetic. You guys okay? So, central nervous system, no subdivisions. It's its own thing. The peripheral nervous system is divided into autonomic and somatic, and then the autonomic is further subdivided into sympathetic and parasympathetic. You guys okay with that? Now the sympathetic branch, I kind of think of the sympathetic branch as the fight or flight branch. Think of this as fight or flight. All right. Fight or flight. Okay, this is the branch. This is the information that is concerned with survival, okay, with immediate survival, okay? So if I get shot or I get stabbed, for example, and I start bleeding to death, it is going to be the sympathetic nervous system that's going to increase my heart rate. It's going to cause glucose to increase in my blood so my cells have more sugar. Okay, it's going to increase my breathing rate. Okay, it's going to cause my pupils to dilate so I can see better, so I can escape. All right. You guys have all experienced a sympathetic response, right? You've been in a car crash, or you've been scared or frightened or anxious or angry. Okay, you've had to run, or you've had to fight, or you thought you might get into a fight. It is this, okay, the fight or flight response. Okay. Now, the parasympathetic nervous system, on the other hand, is what I call the feed and breed. It is the feed and breed. Okay, so this is the branch, this is the information that is primarily concerned with digestion, slowing things down, relaxation, okay, eating, okay. Now, when it comes to the sympathetic and parasympathetic, it's never going to be all sympathetic or all parasympathetic. Don't think of them as antagonizing each other. Okay? What it is it is a balance. Okay? And sometimes you're going to have more sympathetic and less parasympathetic, and other times you're going to have more parasympathetic and less sympathetic. It is a balance. They're not antagonizing each other. They're balancing each other out. Does that kind of make sense? Okay, you guys good with that? All right.
The sympathetic, is that kind of like the example where, like, if you get enough adrenaline, you can lift a car or something like that?
Well, sort of. I mean, when it comes to like lifting and things like that, you can't do more than what your muscles can do.
So that's not really a fight or flight thing.
That is, but as far as like, yes, it is involved in people doing, possibly doing heroic and things like that. But again, it's not magical, and it can't make your body do what your body can't do. Is I guess what I want to say is, yeah. But certainly, it can rapidly immobilize energy. It can increase oxygen levels. You can see better, and it may be involved, and we'll talk about it a little later when it comes to how you perceive pain and things like that. There are some neuromodulators that get released in a sympathetic response where you may be very insensitive to pain. And I bet a lot of you have actually experienced this if you've been in a fight, or like me, I do endurance racing, and several hours in my endurance, it's interesting as generally the first half, like first 5-10 hours of my races are just awful, just suffering, and sometimes I'll even hallucinate when it gets really bad. And then at some point, all that just goes away, and I don't feel any pain anymore, and I end up getting better. I'm moving faster, and I just feel better. And what is that? Well, that's okay. Yeah, that's that. You've all heard of the quote unquote runner's high, right? Those are neuromodulators taking effect and decreasing pain sensation and allowing me to do more than I consciously think is possible. So yeah, it may allow people to do more than they consciously think is possible. And then there's a whole philosophy behind all that when it comes to flow states and things like that obviously I can't get into, but yeah. So you guys okay with that, more or less? Does that kind of make sense? All right, cool. So now what I want to do is I want to move into talking about the cells that make up the central and peripheral nervous system, since we're talking about neurotissues, okay? So the primary functional cell, okay, the primary functional cell is the neuron, right? You guys okay with that?
We're talking about the central.
CNS, PNS, doesn't matter, right?
Oh, okay.
The functional cellular unit of the nervous system is the neuron. And the general, okay, if you look at the general anatomical features of the neuron are such that you have dendrites, and we talked about this in lab, you have dendrites that bring information into the neuron, right? So information comes into the neuron through dendrites, And that information comes and is integrated in what's known as the soma, or the body of the cell. So far, so good. Okay. And then following integration, information is sent away from the neuron via what's known as an axon. And of course, you guys remember talking about this in lab as well, right? So this should be nothing new. And sometimes, and in many cases, the axons are lined in myelin, right? And that acts as an insulator and facilitates the saltatory conduction that we talked about in lab. We'll talk about it here as well. All right. Now, some of the nuance that we didn't talk about in lab, and this is going to be very apparent in this week's lab when we actually go and do the tissue dissection. We dissect the brain. Within the soma, we have dense areas of rough endoplasmic reticulum. And a lot of the proteins and neurotransmitters and such for the neuron are manufactured in the soma, and then they are transported down the axon by kinetic proteins that move, like kinesin is a big one that transports stuff from the soma to the axon terminals. And so you have a lot of activity occurring. You have these dense areas of rough endoplasmic reticulum. And under the microscope, these are known as Niesel bodies. These are known as Niesel bodies, N-I-S-S-L, okay, kind of a weird spelling, Niesel bodies. And when you have many of them together, they have a very dark gray appearance. So when you look at the brain this week in lab, you're going to see areas that are very dark gray in appearance, and we call that gray matter. So when you see gray matter, what you're looking at is you're looking at large concentrations of cell bodies. Does that kind of make sense there? Okay. Now, you also have the axons, which are wrapped in myelin, and myelin's a rather fatty insulating substance, and fat kind of has a whitish appearance. And so you're going to see areas of the brain that appear very white in nature, okay? And that's what we call white matter, and you're actually looking at the axons of many, many, many neurons. So wherever you have white matter, okay, you're looking at myelins, you're looking at the axons, and then whenever you see gray matter, you're actually looking at the neuron cell bodies. Many, many of them, large collection of them. So far, so good. All right. Now, when we talk about the axon has a highly specialized membrane. Okay, because remember that membrane is designed or has evolved to propagate action potentials, right? So it has lots of different ion channels, and so we call that the axolemma. Okay, just like the sarcolemma was the cell membrane for the muscle cell. It's a specialized structure. The axolemma is the membrane of the axon of the neurons, very specialized. And of course it contains what's known as axoplasm, which is just cytoplasm for the axon. You guys okay with that? And then you have this perikaryon. This is cytoplasm within the soma of the cell. Because these neurons are so specialized, like muscle cells, we have separate terminology for some of these things.
That's
Yeah.
the cytoplasm for the soma?
The soma, yeah.
Okay.
The perikaryon is the cytoplasm within the soma, and then the axoplasm is the cytoplasm within the axon. And we have highly specialized structures. For example, the axon, as you might imagine, has highly specialized microtubules. That has a very specialized cytoskeleton, because you've got to be able to transport stuff up and down that axon. Does that make sense? Mm-hmm. So that hence the specialized terminology. Now, once we get toward the end of the axon starts branching out. The large branches are what are known as the collaterals, the axon collaterals. And then the collaterals further branch out into what are known as the teleodendria, okay? The telio or teleodendria, okay, are the fine branches. And at the very end of the teleodendria, you have the actual synaptic terminals themselves, where the of course where the neurotransmitters are stored.
Which those are called, okay, synaptic.
Synaptic terminals. Okay. You guys okay with that? Just some new terminology, really, but shouldn't be too bad. Now what I want to do is I want to talk about the glial cells, and we talked about them in lab, but let's just review them. And we also have to understand that there are different glial cells for the central nervous system and the peripheral nervous system. The central nervous system has some specialized glial cells. The peripheral nervous system has some specialized glial cells, particularly when it comes to producing the myelin, right? So let's talk about the astrocytes. What are the astrocytes? We talked about these in the lab. What are these? Astrocytes. Very, very common glial cell. They do kind of have a star-like shape to them. They have lots of extensions. They have extensions that attach to the neuron and attach to blood vessels.
They keep everything aligned.
They kind of hold, support, protect, and anchor things down, right?
Mm-hmm.
Does that kind of make sense? They also have many other functions that we don't really know, but
They prevent everything from moving.
they kind of move things around. And they may be involved in a highly specialized structure. Okay? It's a highly specialized structure known as the blood-brain barrier. The blood-brain barrier is in essence an extra layer around the blood vessels in the central nervous system.
An extra.
Kind of like an extra layer. Think of it as an extra layer, an extra layering. Okay? It's a specialized structure, and what the blood-brain barrier does is it allows for selectivity. It's even more selective in what can come in and out of the brain. And the blood-brain barrier is sometimes a big problem for us, because when we have problems with the brain, okay, for example, one problem is a disease known as Parkinson's disease. You guys have all heard of that. And one of the problems with Parkinson's disease is you have a decrease in a certain neurotransmitter in a certain area of the brain known as dopamine. Well, dopamine is a very common drug that we give to people. We can give it intravenously, right? It's a very common drug. The problem is, and so you think, well, why don't we just give people Parkinson's disease dopamine, right? That's what they need. Just give it in the IV, it'll circulate to the brain, and we'll be good to go, right? Well, wrong. Problem is, dopamine can't get through the blood-brain barrier. So you can put that dopamine in the IV and it'll get circulated around, but it won't be able to get into the brain where it needs to go because of the blood-brain barrier. If you have an infection in your brain, it can be very hard to treat brain infections because a lot of the antibiotics that we have are not able to get through the blood-brain barrier very effectively. So that blood-brain barrier is protective, right? It allows the brain to be very specific about what comes in, what comes out, but it is also problematic when it comes to using medicines to treat disorders. Okay, so those are the astrocytes. What about the microglia? Those tiny little star-shaped things that surround the neurons. What's going on with the microglia? What's their role? What's their function? We think might be their function.
Get rid of the waste.
So yeah, they do have kind of a protective, they're kind of an immune-like function. Okay. They may be able to get rid of things. They may be able to kind of clean things up after an infection or trauma or inflammation. The astrocytes may do a little of that as well. Okay. What about the ependymal cells? What are the ependymal cells? What's their job? What What are these?
They're in the CSF.
Yeah, so they line the ventricles. They line cavities within the central nervous system. Remember we talked about them in lab, and they have cilia, and they produce the cerebral spinal fluid, right? The CSF. They produce and circulate the cerebral spinal fluid, which acts as an extra protective cushioning for the brain and spinal cord. Good. All right. And sometimes too much cerebral spinal fluid gets produced, and that causes a problem known as hydrocephalus. Sometimes that's a problem in babies, and we actually have to put a shunt. We actually have to put a tube in their brain, and then that tube goes down into one of their large blood vessels to help drain all that extra CSF off that gets produced. Okay. And then finally, the other major one I want to talk about are the oligodendrocytes, and what do they do in the central nervous system? Very important function. They produce something very important.
Myelin?
The myelin. But only in the CNS, right? Only in the CNS. Now, the peripheral nervous system has specialized glial cells as well. Look at that, I even color-coded it for you guys. Hopefully no one here is colorblind. I actually have had students that are colorblind. This didn't really help them very much, but oh well. You have satellite cells and Schwann cells. Okay. Specifically, what do the Schwann cells do in the peripheral nervous system? So you definitely need to know this. What do they produce in the peripheral nervous system?
The Schwann cells?
Yeah, right. PNS.
Myelin.
The myelin. Yeah. All right. Again, this is peripheral nervous system, right? And CNS. And remember, in the peripheral nervous system, one Schwann cell, one piece of myelin, one neuron, right? In the CNS, an oligodendrocyte can have multiple extensions and produce multiple myelin linings around multiple neurons. So they're highly specialized, and they're a little different in how they work. So far, so good?
So the Schwann cells have like a kind of 1-1 ratio, but
Pretty much.
the oligodendrocytes don't.
Don't, yeah. Guys cool with that? All right, so let's move on to talking about the types of neurons. And we can categorize neurons in several different ways. So the first way that I want to categorize is generally by how they look. Okay? The classic neuron, okay, the classic picture that everybody sees, is what's known as a multipolar neuron. Okay? And this is a neuron that has multiple dendrites coming into a soma, and then an axon leaving that soma. Okay? This is the classic picture that you've all seen of a neuron. Okay? These are very common. They are found in both the central and the peripheral nervous system. And one of the major jobs that multipolar neurons are involved in is with motor, is sending motor information out to your muscles. Okay, if you're talking about the somatic nervous system, and sending motor information out to glands and smooth muscle if you're talking about the autonomic nervous system. Okay, so those are your multipolar neurons. Another type of neuron is what's known as a bipolar neuron. These tend to be very rare, however. These tend to be very rare, and we only see them with special senses, such as olfaction. Olfaction also means to smell, and vision. Olfaction, vision. Okay, so those nerves that detect the vision, detect sense of smell, and send that information to the brain. Those are what we call bipolar neurons. And basically what you have is you have the soma in the middle, and then you have just a little collection of dendrites here, and then you have your axon that goes into the soma, and then an axon that comes out with axon terminals. Okay? So basically, if I were to move this soma into the middle of the axon, I would, in essence, convert my multipolar to a bipolar neuron. You guys okay with that?
And it ends with the dendrites again.
No, axon.
Dendrites? Oh.
This is the axon terminals here, the synaptic terminals. I just simplified the picture. The next one, which is actually a little more common than the bipolar, are what are known as unipolar, or really what they are is they are pseudo-unipolar. They appear to be unipolar, but they're not quite. They can go by both names, either unipolar or pseudo-unipolar. And in this particular neuron, the cell body is separate from the axon. Okay, so you can see there's just a little line that connects it to the axon there, but the axon is just kind of hanging off the cell body, and it's all by its lonesome, more or less. You guys see that there? And what you typically have with pseudo-unipolar neurons are you have your dendrites. Okay, the dendrites are in the peripheral nervous system. Okay, and then you have your axons in the central nervous system, or sometimes vice versa. Okay, where do we see these? These are some of the most common sensory neurons. These are the neurons that carry a significant amount of sensory information into the brain, or the afferent pathways, with the exception of special senses. Special senses are bipolar neurons primarily, vision, olfaction, okay? But all of your other senses, okay, tend to be the unipolar or pseudo-unipolar. And then finally we have what are known as anaxonic neurons, and these are neurons that don't really have an identified axon. They kind of are star-shaped and they have multiple dendrites coming into them. Okay? We see these primarily within the central nervous system, and sometimes we see them in very specialized sensory organs as well. So far, so good? So this is looking at the neurons by their morphology, by what they look like. Okay, your multipolar, your bipolar, your pseudo-unipolar, and your anaxonic. We can also look at neurons by what they do. Okay, and there are three general jobs for a neuron. Okay, this is what a neuron does when it gets up in the morning and goes to work. Okay, it can either be a sensory neuron, a motor neuron, or it can be an interneuron. Okay, so sensory, motor, and interneuron. You guys hanging in there okay? Mm-hmm. All right, excellent. So let's talk about sensory neurons. What do sensory neurons do? They monitor both the internal and external environment. Again, these are typically going to be unipolar. Okay? And they are afferent, right? They send information up to the central nervous system. And they are typically, okay, associated with receptors, with sensory receptors, of which you have three major types. These are not special senses though, okay? So when we talk about vision and smell and taste and sight and sound, the special senses, we're going to have a whole lecture on special senses. These are your run-of-the-mill, if you want to look at them, these the everyday senses, okay? So you have your interoreceptors, okay? Or your internal receptors. These are within the body, right? So these monitor things like blood pressure, temperature, pH levels, things like that. You have your exteroreceptors, and these are receptors that monitor the outside environment, okay? So is there a break in the skin, okay? Or, do you have a pain receptor? And then you finally have what are known as proprioreceptors. And what do proprioreceptors monitor? They monitor our sense of spatial orientation and movement. Okay, so this is information about where I'm at in space. How am I oriented? Am I standing? Am I sitting? Am I lying down? Am I upside down? Am I spinning around? Okay, what we call your proprioception your general sense of balance. You guys okay with that? All right. So these are your sensory neurons. Now, your motor neurons, on the other hand, okay, can either be somatic or autonomic, right? And we're talking about the peripheral nervous system here. And remember, the motor neurons tend to be multipolar. They tend to be that classic neuron that we all know and love, we've all seen. Okay? Remember, somatic tends to be voluntary skeletal muscle, right? Voluntary control. Autonomic, on the other hand, tends to be involuntary. It tends to involve smooth muscle, okay? It tends to involve organs, and it tends to involve glands, right? So like salivation, okay? Salivation, lacrimation, glands that secrete mucus in your airways, smooth muscles, such as smooth muscle in your lungs, your intestines. All of that is autonomic. And what we tend to see there, okay, with autonomic, is we tend to see a neuron that leaves the central nervous system, okay? It leaves the central nervous system, and then it synapses with another neuron that then brings the information to the gland, organ, smooth muscle, or what have you, okay? So it's a two-neuron system. So your autonomic branch tends to be a two-neuron system. The initial neuron that leaves, okay, so the initial neuron as it leaves the CNS, and then it ends, and it synapses with another one, okay? And those synapses tend to occur in groups. You actually have groups of these synapses, and these are known as ganglia, or ganglion if you're talking about just one. Okay, ganglion, or ganglia, being plural, are just large groups where you have cell bodies and you have synapses occurring, okay, between the CNS, okay, coming from the CNS and going away from the CNS. And what we tend to do is we tend to say neurons that are going into the ganglion are known as preganglionic neurons. They're coming into the neuron. And then neurons that are leaving the ganglion to bring information out to the organ, tissue, gland, what have you, are known as postganglionic. Does that kind of make sense there? Excellent. And then finally, we have what are known as interneurons. And these are the in-between people. Okay, these relay information between the sensory and the motor neurons. Because how do I connect the sensory motor, right? Up to this point, we've just said that sensory information comes in, motor information goes out, but something has to happen, right? The sensory information has to be integrated, has to be processed, right? We have to make sense of the sensory information to send that motor information out. And that's what these interneurons do. They relay, or they're also known as association neurons. Okay? They are a way of communicating between the motor and sensory. And so what I want you guys to do is I want you to imagine and this is a classic example here, what we call a reflex arc. A reflex arc. You guys are familiar with reflexes, right? Okay. So imagine that I've got a fire here. Yes, it's supposed to be a little fire. Okay. And I'm having a good time out camping or whatever, and I forget there's a fire there, and my hand, that flame comes over my hand. Okay. And so what's going to happen is that flame is going to activate exteroreceptors, right? Receptors in my skin that are monitoring, hey, it's getting kind of hot here. We're taking some damage, right? And so what those receptors are going to do, they're going to send sensory information, okay, through an afferent pathway into the spinal cord, right? And the spinal cord is a CNS. Now here's the cool thing about a reflex. A reflex does not need to occur in the brain. So imagine that my hand's getting burned, or it's getting stung or stabbed or poked or what have you. If my hand or part of my body is getting damaged, is it advantageous to spend a lot of time integrating and processing that information? No, right? So I need a way of responding to that damage very quickly. And so what we actually have is, as that sensory information comes in, before it even goes to your brain, okay, so before you are consciously aware that you're taking damage, that information will get processed by an interneuron within your spinal cord. Okay, so the sensory information comes up, it goes into the interneuron, there's a synapse in the spinal cord, and the interneuron then sends motor information out through an efferent pathway to the muscles in your arm and causes them to contract, and you pull away from the fire. Okay? And that's what we call a reflex, or an interneuron in the spinal cord. Okay, makes the connection between that sensory information coming in and the motor information going out. And actually it kind of makes sense that we would want to have these, right? And you guys have probably all had a situation where you've been burnt, or you hit your hand with a hammer or something like that. And what did you notice before you even felt pain? Before you were even aware that you were hurting? What did you do? Huh? You pulled away. Ah! And then what happened? Ow! Ow! Then you started feeling the pain. But you reacted before, right? Before you actually experienced pain. Why is that? Because this reflex happened right away, and then of course sensory information finally makes its way up to the brain. Interneurons in the brain then process it and go, oh, we're having some pain here. And then you start experiencing pain. Does that kind of make sense there? Yeah.
Can reflexes be created over time, or is it something that's already there?
Yeah, reflexes are generally kind of pre-existing. I don't know how much they can be developed.
Like,
They might be, because these are interneurons. So
would that just be like muscle memory neurons?
yeah, muscle memory might be, and I don't know how much they can be developed. How much the unconscious kinds of reflexes can be developed.
Like for anything, someone tries to punch you, what are you gonna do?
Yeah, you're gonna pull away. Yeah, you
or
see
like
that movement.
For a fighter, you do
Yeah.
something different, you know what I mean? But that stuff comes as second nature, not like, it doesn't process like, oh, I'm gonna do this.
Right, it's not a
So is
conscious
that-
thing.
Yeah, but
Yeah.
when I was a kid, I never had those skills, you know what I mean? So is that something that can be developed, or would that just be more or less muscle?
Oh yeah, as a kid, yeah. Because as a kid, you're born with your nervous system is not developed when you're born.
Well, no, that's what I'm saying, can you like develop those, can it be developed, you know what I mean?
As an
Like,
adult?
now, that would be something that would be a reflex to me. As a kid, it wouldn't be, but it would still, as a kid, it would be a reflex to if I had something hot. You know,
Right,
if I-
yeah,
That would be
yeah.
a reflex
Uh-huh.
my whole life, but as a kid, if someone punched me, I probably would have just stood there, you know what I mean? But now, obviously, that's not the case.
Right, yeah.
Like,
Oh yeah. These develop for sure. Just through, particularly as a kid, because you don't have your nervous system is not fully developed, and in fact, your nervous system doesn't fully develop until you're well into your 20s. Like people are generally do not have a fully developed nervous system until they're well into their 20s.
You can train reflexes for you.
I don't know how much they can be consciously trained. I don't really
Not
know.
consciously trained, but like after so many times of doing it, some like things just come second nature as a reflex.
But I don't know that that's due to the interneurons in your spinal cord.
Versus muscle memory.
Yeah, so much as neurons here up in the higher centers of your brain.
Would that be more of like a process of pruning? So, when you're younger,
You do
your
undergo
synapses
pruning, yeah.
become a lot more centralized
Mhm.
to like the use it or lose it thing.
Yep.
Would it be more related
I mean, you have more synapses as a kid than an adult, and then what happens is you kind of start solidifying your development. They kind of those they get pruned. You know, they cut them away. They kind of go away. Yeah. But yeah, you don't have full neurodevelopment really until you're well into your 20s. It's kind of an interesting thing to think about. And that is part of the when we talk about like the drinking age, things like that. And there are some places that are actually looking at possibly making the drinking age or the smoking age now. They're possibly thinking about making it 21 instead of 18. And some, I won't say all, but some of the rationale is, well, you still have, your brain is still developing in your 20s. So maybe there's some possible problems there, but there we go. All right. Kind of makes sense a little bit there? Okay, so interneurons, that's just, the reflex arc is kind of a classic example of an interneuron at work in the spinal cord in this case. Okie dokie, how are we doing? Okay, I'll go for another few more minutes and get you guys on a break. So now what I want to do is I just want to go back and I want to review how this whole action potential business works. We talked about this in the lab. Okay, so we said that neurons are an all or nothing kind of deal, right? A neuron will either fire or it won't fire, right? It's all or nothing. There's no half measure here when it comes to a neuron. So what we have happening with neurons is neurons have the sodium-potassium pump in their membrane, right? And that sodium-potassium pump actively transports sodium out of the cell and potassium in. And within the cell, you actually have negatively charged proteins, okay, or these anionic proteins. So with having all the sodium out, negatively charged anionic proteins inside and some potassium inside, what we do is we can compare the inside of the cell, the outside of the cell, and what is the inside of the neuron when we compare it to the outside. What kind of charge do we have? Charge difference do we have going on? It's much more negatively charged inside versus outside. And so we say that the membrane potential, okay, the potential across that membrane is negative, you know, negative 90, 110 millivolts, somewhere in there, okay? Just like a muscle. It's very similar to a muscle cell in that sense, okay? And so what we say is that the resting potential, okay, when the neuron isn't doing anything, okay, is very negative. Does that kind of make sense? And then what can happen is there can be information that comes into the neuron, and we call these graded or local potentials are produced. So remember, those neurons have thousands of dendrites, and some of those dendrites, excitatory information is coming in, right? And some of those dendrites, inhibitory information is coming in. And excitatory tends to be sodium ions flowing in, right? Something that causes sodium to go into the cell. And so what happens is, if enough sodium enters the cell to get that membrane potential up to the threshold, what happens?
Action potential.
A full-on action potential. Okay, if the sum, right? If the sum of the excitatory and inhibitory input, okay, can get you up to the threshold, those voltage-gated sodium channels in the axon hillock there will open up. Sodium will rush into the neuron, and the neuron will undergo depolarization. Does that kind of make sense? So it is this balance of inhibitory and excitatory information coming into the neuron, and if the inhibitory information exceeds the excitatory information, the neuron will not depolarize, right? But if the excitatory information exceeds the inhibitory information coming in, the neuron will depolarize as long as you can get above that, at or above that threshold, right? And of course, that's something known as temporal summation. And actually that sigma there, that large sigma, that just is a symbol that means to sum up, right? So it's the sum of the excitatory and inhibitory information coming in. If you get up to the threshold, the voltage-gated ion channels open up, and sodium rushes into the cell, and it depolarizes and rapidly becomes positive, right, as more positive ions flow in. Now, what happens is, right here at the hillock, as that sodium rushes in, there's a lot of sodium right here, right? It's very concentrated. And then as that signal passes through the axon, the sodium kind of gets diluted out, right? It spreads out. And so that signal, okay, kind of spreads out a little bit, doesn't it? And it weakens until you get to a node of Ranvier, right? Because remember, this is surrounded in myelin here, right? And once I get to a node of Ranvier, there are sodium channels, voltage-gated sodium channels that open up. Sodium rushes in, the signal then becomes stronger, and then it spreads out again, stronger, spreads out, stronger, spreads out, and it does this until eventually the signal gets to the axon terminal, the end of the axon. And so this stronger and then diluting out, stronger diluting out, kind of creates a hopping kind of signal, and that's of course what we call the saltatory conduction, right? You guys okay with that?
So the node of Ranvier brings the sodium in still from outside the membrane?
Yes.
Okay.
Yeah, so you have voltage-gated sodium, and it basically amplifies. Basically, the signal gets amplified at the node of Ranvier. Right? Just like if you had a 100-mile electrical cable. If I had a 100-mile electrical cable and I plugged it into the computer and then 100 miles away was the electrical outlet, do you think the computer would work? No, because that electrical signal would get very diluted out, for lack of better words.
So what if the voltage-gated ion channels never opened up? What
Yeah.
if there the sodium still coming through the
No.
node of Ranvier?
No.
So why is there sodium coming in from the node of Ranvier? Is there a voltage gated?
Yeah, there is a voltage.
Okay.
Yeah, there's a voltage-gated channel. There are many channels in the nodes. Yeah, and sodium, like I said, Drew, this sodium. So sodium rushes in, the signal gets amplified again, and then dilutes out, amplified, dilute out, amplified, et cetera, et cetera. That kind of makes sense there.
Yes.
Right? So that's what we see with myelinated axons, at least. And then that depolarization gets to the axon terminal, okay? And then there are voltage-gated calcium channels here. And the voltage-gated calcium channels sense that change, sense that sodium coming in. They open up, calcium rushes into the axon terminal, and calcium is what enables these vesicles containing neurotransmitters to leave the neuron. And then the neurotransmitters get dumped out into the synapse here, right? And then post-synaptically, the neuron or other tissue, okay, that's on the other end of that synapse will have receptors that those neurotransmitters will act on. And ultimately, those neurotransmitters will either be excitatory or inhibitory. And then the whole thing starts again. Does that make sense? All right, it's exactly an hour. Let me get you guys on a little break. I'll see you guys in about 5 minutes, and we'll